Hello everyone. In this video lecture, we will talk about prions, the infectious proteins responsible for mysterious diseases like Kuru, Scrapie, the mad cow disease, the CJD. So stay along to unravel the mystery of these prions. You have probably heard about the mad cow disease outbreak that occurred in England a few years ago. Mad cow disease or bovine spongiform encephalopathy, as it is scientifically called, is a deadly disease of the cattle's central nervous system. Small holes develop in the animal's brains, making them eventually look like big hole-riddled sponges. Dozens of people in England are thought to have acquired a human form of BSE by eating the meat of the infected cattle. The human illness is called a new variant Creutzfeldt Jacob disease, the new variant CJD. A similar disease is seen in sheep and goats. The disease is called Scrapie. The animals become so uncomfortable and itchy that they frantically rub up against anything they can, finally scraping off most of their wool and hair. Hence the name Scrapie. Let's now talk about Kuru, the first human prion disease. Kuru is the name of a similar condition found among members of the Kuru tribe in the Four Highlands of New Guinea. The disease was the result of the practice of ritualistic cannibalism among the Four, in which the relatives prepared and consumed the tissues, including brain, of the deceased family members. This was probably the mode of the transmission, as a result of which all the members of that tribe suffered from the disease to the extent that the tribe got completely extinct. All these diseases have two things in common. First, they are all fatal neurodegenerative diseases, together referred to as transmissible spongiform encephalopathies. Transmissible spongiform encephalopathies are a family of rare progressive neurodegenerative disorders that affect both humans and animals. They are distinguished by long incubation periods, characteristic spongiform changes associated with neuronal loss, and a failure to induce inflammatory response. These diseases are also characterized clinically by motor disturbance and dementia, and pathologically by neuronal vacuolation, astrocytic gliosis, and amyloid plaques. On your screen, you can see a normal human cerebral cortex staining, and this is spongiform encephalopathies characterized by amyloid plaques or spongy appearance. You can also see the neuronal vacuolation. You can see this vacuole which is shown here with an arrow. The second thing common to all these disorders is that they are all prion disorders. The main histopathological marker of TSEs is the cerebral accumulation of a misfolded form of the cellular prion protein PRPC, designated PRP-SC. Here C refers to cellular or PRPC and SC refers to Scrapie, a prion disease occurring in the sheep. Let us now talk about the discovery of prions. Prions are infectious proteins devoid of nucleic acids. They were discovered by Stanley Prusiner in the year 1982, for which he was given the Nobel Prize in the year 1997. Prusiner's interest in the subject began with a patient dying of CJD in the fall of 1972. At that time, he was beginning a residency in neurology and was most impressed by a disease process that could kill his patient in 2 months by destroying her brain while her body remained unaffected by this process. He was told that she was infected with a slow virus. His fascination with CJD quickly shifted to Scrapie. This was because he came to know that the Scrapie agent was extremely resistant to inactivation by UV and ionizing radiations. Also, that Scrapie agent could be inactivated by procedures that hydrolyze or modify proteins. These data established for the first time that a particular macromolecule was required for infectivity, and that macromolecule was probably a protein. Once this was established, Prusiner thought of giving the infectious pathogen of Scrapie a name so as to distinguish it from viruses and viroids. He suggested the term prion, which was derived from proteinaceous and infectious. So prions can be defined as proteinaceous infectious particles which lack a nucleic acid. Talking about the isoforms of prion protein, the prion protein responsible for Scrapie infectivity was designated PRP-SC, coded by the PRP gene. The protein on subjection to proteases gave a protease-resistant core of 27 to 30 kilodaltons. This was designated PRP 2730. It was found to be insoluble in a non-denaturing detergent. Thus, PRP-SC was found in all cases of Scrapie infection. But surprisingly, the PRP mRNA levels were found to be similar in both normal and Scrapie infected tissues. This led to the search of a prion protein in uninfected tissues, which was probably protease sensitive and soluble in non-denaturing detergent. This isoform of prion protein, when purified, was designated PRP-C, which indicates PRP cellular. Thus, there are three isoforms of the same protein: PRP-C in uninfected cellular tissues, PRP-SC in infected tissues, and PRP-2730, the protease-resistant core. Once cDNA probes for PRP became available, PRP gene was found to be constitutively expressed in adult uninfected tissues. Also, the PRP gene was found to have a single exon. Thus, there was no possibility of two PRP isoforms to be products of alternatively spliced mRNAs. It was also contemplated that PRP-SC must have been derived from PRP-C as a result of post-translational chemical modification, but none was found. Thus, the only possible explanation was that PRP-SC and PRP-C differed only in their conformation. It became evident from FTIR and circular dichroism studies that PRP-C had 40% alpha helix and as little as 3% beta sheet, whereas PRP-SC contains 30% alpha helix and 45% beta sheet. But the primary structure, that is the amino acid sequence, was exactly the same. Thus, a comparison between PRP-C and PRP-SC can be made. PRP-C is a normal cell surface protein, whereas PRP-SC is present only in diseased brain. PRPC is not infectious, whereas PRPSC is an infectious protein, that is, it is a prion. PRP cellular is protease sensitive, whereas PRP Scrapie is protease resistant. PRPC is monomeric, whereas PRPSC is aggregated. PRPC, as we just saw, is 3% beta sheet and 40% alpha helix, whereas PRPSC is 40% beta sheet and 30% alpha helix. The PRP gene. The gene coding the cellular isoform of prion protein PRPC is located on the short arm of chromosome 20 in humans and is called the PRP gene or PRNP gene. It is present as a single copy gene in the genome and is constitutively expressed in the adults. Let us now talk about the cellular prion protein PRPC. The PRPC protein encoded by the PRP gene is approximately 250 amino acids in length. It has an amino terminal signal peptide and a hydrophobic carboxy terminal domain for membrane attachment via glycosyl phosphatidyl inositol, that is GPI anchor. There are 4 copies of an octapeptide repeat with close analogy to a proximate nonapeptide in the N-terminal region. There is a single disulfide bond between the cysteines 179 and 214 and 2 N-glycosylation sites at asparagines 182 and 198. A hydrophobic sequence in the middle of the protein may exist as a transmembranous region in some PRP isoforms. Let us now discuss the physiological function of PRPC. The precise function of PRPC is unknown. However, considerable progress is being made towards understanding its physiological activities. PRPC has been associated with maintaining cell survival, promoting neurite outgrowth, modulating synaptic transmission, and copper homeostasis. It is a well-known fact that copper imbalance causes dysfunction of the central nervous system. The structure of the PRPC shows that there are 4 octarepeat regions which bind to copper. Probably, PRPC acts like a copper transporter which maintains a balance of copper inside the cell. When PRPC gets misfolded to PRPSC, it loses its ability to bind copper and probably leads to copper imbalance. It is also supposed that PRPC is an antioxidant. Once it binds to copper, it may have an activity like superoxide dismutase. Conversion of PRPC to PRPSC leads to a loss of antioxidant protection that could be responsible for neurodysfunction in the disease. Talking about the prion pathogenesis, infection of normal cells may occur when PRPSC acts as a template for the refolding of a normal prion PRPC into a new aberrant PRPSC. It is thought that there is at least one more protein involved, the as yet unidentified protein X. This protein is believed to mediate the folding from a normal into an abnormal prion. It has been hypothesized that protein X is a type of a chaperone protein. These PRPSC accumulate intracellularly in cytoplasmic organelles and in the extracellular space in form of amyloid plaques. But the question which is unanswered is the accumulation of PRP-SC in brain cells enough for pathogenesis, which includes symptoms like vacuolation and eventually death of nerve cell, activation of astrocytes and microglial cells, and lethal impairment of electrical functions of cells? Let us now discuss the models for the PRP-C to PRP-SC conversion. Among the various mechanisms proposed for the PRP-C to PRP-SC conversion, the two most often considered are the heterodimer or template-assisted model and the nucleation-dependent polymerization model. The first, heterodimer template-assisted model. The template-assisted model proposes that the PRP-C to PRP-SC conversion is a kinetically controlled process. According to this model, the PRP-SC form is inherently more stable than PRP-C but kinetically inaccessible. In this case, PRP-SC could promote conversion by catalyzing the rearrangement of a molecule of PRP-C or of a partially destabilized intermediate to the more stable PRP-SC conformation. The rate-limiting step for the conversion reaction in this model is the formation of a PRP-SC monomer that can then bind to a PRC molecule, thus generating a heterodimer. Subsequently, PRP-SC promotes the rearrangement of PRP-C to PRP-SC, leading to a homodimer that further dissociates into two PRP-SC monomer molecules, as can be seen in the figure on your screen. In this mechanism, the formation of the ordered oligomers occurs after the conversion. The second model is the nucleation-dependent polymerization model. The nucleation-dependent polymerization model proposes that the conversion between the monomeric PRP-C and PRP-SC is a fast and reversible process, but PRP-SC monomer is less stable than PRP-C. The formation of a PRP-SC aggregate is required, which promotes the conversion of PRP-C by binding to and stabilizing the otherwise unfavored PRP-SC conformation. The rate-limiting step is in the PRP-C to PRP-SC conversion is the formation of an oligomeric PRP-SC nucleus or aggregate, which promotes the reaction by further incorporation of monomeric PRP-SC. After having discussed several features of prions, let us quickly revise a few characteristic features of prions. The prions are proteinaceous infectious particles. They are abnormal proteins that have an affinity for the central nervous system. They are very highly resistant to heating. They are not sensitive to irradiation. They are not destroyed by enzymes that damage DNA or RNA. They are sensitive to protein denaturing agents, however. Abnormal prions contact and change the shapes of other normal prions in the nerve cell, and the nerve cell then tries to get rid of them. When the cell dies, the abnormal prions are released to infect other cells. Large sponge-like holes are left where many cells. Let us now talk about a very important question. What is the reason of prion disease? Is it deficiency of PRP-C or production of PRP-SC? The protein-only hypothesis proposes that PRP-SC, when introduced into a normal host, causes conversion of PRP-C to PRP-SC. Therefore, an animal devoid of PRP-C should be resistant to prion diseases? It has been observed that mice develop normally despite complete absence of PRP-C. The PRP knockout mice were viable and remained free of Scrapie for 2 years after inoculation of prions. Also, there is no multiplication of prions in PRP knockout mice, but there are reports of a different strain of knockout mice developing ataxia by 70 weeks of age associated with loss of cerebellar Purkinje cells, further supporting the idea that PRP deficiency could promote neuronal cell death. The other important question was, can production of PRP-SC alone produce disease? An elegant study of Agusi, Weissman and co-workers has addressed this question. They engrafted brain tissue from a PRP-C expressing normal mouse into a knockout animal and then inoculated with mouse prions. They observed the characteristic pathology of prion disease precisely within the engrafted tissue, but not within the knockout tissues. Nevertheless, PRP-SC produced within the diseased tissue migrated into the neighboring null tissue. Thus, PRP-SC by itself was insufficient to produce the prion disease. Simple exposure to exogenous PRP-SC is insufficient for cytotoxicity. Rather, PRP-SC may be toxic only when formed within the cell or when presented from outside to a cell already expressing the cellular PRP-C, so that conversion ensues either at the cell surface or in an internalized compartment. Thus, in all cases, it appears that the presence of PRP-C, that is the cellular PRP protein, is required for pathogenesis. After having discussed the prion pathogenesis, let us now move on to the treatment of the prion disorders. PRP overexpression facilitates the development of prion diseases. It may therefore follow that agents which reduce PRP expression will delay the onset of prion diseases. One can speculate that chemicals which bind to and stabilize the PRP-C conformation may be beneficial. Similarly, agents destabilizing the PRP-SC conformation may be effective. In this regard, several vaccines to Alzheimer's amyloid plaques are in clinical trials. Agents which interfere with the putative PRP-C to PRP-SC interaction might similarly be effective. A number of reagents showing affinity for amyloid proteins are known, for example, Congo Red. As our knowledge of the structure of PRP increases, the chances of rationally deducing effective therapeutics based on these ideas also increases. Finally, we have seen that PRP expression is required for pathology. Chemicals affecting the endocytosis, exocytosis, intracellular trafficking, and degradation of proteins, and in particular PRP, may also be effective. Amphotericin, for instance, is reported to delay prion diseases in hamsters, although it apparently has been found to have very little effect in humans. After having discussed so much about the prions and prion diseases, there are many questions about prions which are still unanswered. An enormous body of data reveals the central role of PRP protein in a group of related transmissible neurodegenerative diseases. These data demonstrate that PRP protein is required for the disease process and that the conformational conversion of the PRP protein from its normal soluble helical conformation to an insoluble state is intimately tied to the generation of disease and infectivity. Much about the conversion process, however, remains unclear. In particular, is introduction of the PRP-SC protein alone sufficient for infection in presence of PRPC? What is the nature of the conformational change? Given the ubiquitous distribution of PRPC, why is prion pathology restricted to the central nervous system? What determines the species barrier? In addition to further resolving a fascinating biological process, the exposure of both cows and people to BSE underscores a need to answer these questions. With this, we come to an end of our discussion on the prion proteins and the disorders which are caused due to prion proteins.